Very good afternoon to all. Welcome back, everyone, and thank you for being with us. Before the break, we had listened to two absolute legends of the field, and now we have one more legend for you for another interesting and informative session. As we head on to the eighth technical session, I would like to call Dr. Uttam Mandal to introduce our speaker, Dr. Yogeshwar Bachchan. Over to you, sir.
Dear participants, afternoon, and I once again welcome you to the last technical session of the day. Dear participants, it is said, a friend in need is a friend indeed. So this is how we can best describe our speaker of the day, Dr. Yogeshwar Bachchan, who happened to be my best friend. First time we met each other 12 years ago in 2008 in Switzerland, while we were working at University of Geneva, Switzerland, under Professor Y.N. Kalia, a renowned researcher in transdermal drug delivery. Since then, we remained in touch, and whenever I need for any kind of help, be it technical help in my research or in personal life, he is always there with his helping hand. So it gives me immense pleasure to introduce such a nice friend to you, dear participants. Dr. Yogeshwar Bachchan basically is from Maharashtra. Did his PhD from ICT, that is Institute of Chemical Technology, Mumbai. After my postdoc, I returned back to India. Well, while Dr. Yogeshwar Bachchan continued working in Europe. And dear participants, let me tell you, this has been possible because of his in-depth knowledge in his subject of drug delivery and his linguistic skills. You will be astonished to know that within a short span of time, he mastered two difficult language, which is French and German. He is an example of perfect blend of profound scientific knowledge and literary talent. He worked in Europe for around 14 years with different startups and mid-sized biotech and pharmaceutical companies. During this tenure, he has successfully managed pharmaceutical development of several projects and worked in phase 1, phase 2, and phase 3 clinical trials. He has strong network of experts and CROs in Europe, USA, and Australia. In addition, he has 16 international publications, 8 patents, and 26 conferences. Very recently, he has edited a book for Wiley in the Science. The title is Innovative Dosage Form, Design and Development at Early Stage. He is founder of his own pharma consultancy firm, which is Adex Pharma, that was established associate director at AI Curis Anti-Infective Cures GmbH, a pharmaceutical company in Germany. So with this short introduction, I welcome Dr. Yogeshwar Bachchan as our expert for this session. Today, he will be talking on a topic entitled Parenteral Nanomedicines Consideration in Developing Complex Nanoformulation. So with this short introduction, I welcome Dr. Yogesh. Dr. Yogesh, it's over to you.
Okay. Good afternoon all. First of all, for this introduction, I think he's more an elder brother for me. So it's equally whenever I needed him, he was always there. So I think it's a great pleasure for me to be here, and I think I'll be very happy if little knowledge from my side, what I have gathered, during my tenure, the different pharma companies in Europe, if you can benefit from today's talk, I think that's the biggest satisfaction I can have. So I think Uttam Dada has already given some introduction, but maybe I'll just give some specific details so that at least the overall aim is you understand what is my background, what you can expect from me, and how I have tried to give justice for today's talk. So as Dada said, I started, I did my PhD from ICT Mumbai. There I mostly work with the oral and topical formulations. Then I moved to Dr. Yogi Kalia's lab in Geneva, where I was working with Dada. There we developed a transdermal medical laser device for the skin delivery of peptides, proteins, antibodies. So it was sort of non-invasive delivery of macromolecules, because for proteins delivery normally you need a IV injections, or maybe let's say parenteral medication, which could not be very practical when it is a chronic administration, especially for in vitro fertilization. You need 90 days of daily injections, and this is a combination product, tryptolin, follicle stimulating hormone, and progesterone. So overall aim was, we tried to create the pores on the skin by using the laser device, and then we tried to apply the patch, and aim was to get a pregnancy by using this transdermal laser device rather than using the 90 days of parenteral medication. And I think I'll be happy to share, when I led that project, that time they had first successful pregnancy from the clinical trial in Czech Republic. So that's where we got feeling how the proof of concept works. So the laser device we were working, it was literally a table shaped device. These days, if you see the website of Pantech Bioengineering, they have a handheld device like a TV remote, and that's how the transition took place. But I think that gave enough zeal of research and how it works. Then I started working with Debio Farm, Swiss biotech company located in Lucerne, as a formulation manager. Overall aim was to develop the preclinical clinical formulations for the oncology pipeline. And these are like new drugs, so new chemical entities coming from preclinical trials. You develop it all the way from the phase 1, phase 2, and phase 3. There I work around 4 years. Quite interesting experience. There I work with oral, topical, as well as parenteral formulations. And then I moved to Germany. So Icarus is basically the bear skin of. I started working as a head of pharmaceutical development in the year 2014. Again, it was like, let's say the biotech company is 60 people with big responsibility of managing clinical trial supply to develop the preclinical clinical formulations for the new drugs in the anti-infective segments. In 1 year time, family was moving back to India for some reasons, but I had to continue. So there we had a solution. Then I could be like full-time consultant to the German company. That also allowed me to work as a consultant to some Asian companies, where I founded this Adex Pharma, and it's working quite well. So I'm still active at Icarus as a full-time consultant, which takes at least 90% of my time. Around 10 to 15% I try to contribute for other consultancy projects in India. So in a nutshell, that's my, let's say, more elaborate introduction. So in case of course you have some other questions than today's talk, I'll be happy to help. So with your due permission, I'll share my screen, and then at least I can talk, start my talk. Can you see the presentation?
Yes, Dr. Yogesh.
Okay, good.
We can see you.
Okay, perfect. I don't know if I can, okay. Okay, so the topic of today's talk, parenteral nanomedicine, and then what consideration we should take while we are developing complex nanoformulations. So what are the prerequisites when it comes to the parenteral formulation? It must be safe, efficacious, because you don't want to have any toxicity from that at least for 24 to 36 months at room temperature, or maybe in some cases 2 to 8 degrees Celsius. And you don't want to have any intolerance issue at the site of injection. And this is to be considered for any formulation scientist who is aiming to develop the parenteral medication. These are very important aspects. And on the right hand side, you see some details. Of course, you don't want to have any hemolysis. Some drugs are known to have hemolytic potential that you need to take care. If your formulation is iso is not having osmolality, which is matching to blood's osmolality, or it should be like what you can say iso-osmolar. Should not be hyper or hypo, or otherwise you may land into this hemolytic potential. As said before, you don't want to have any pain at the site of injection. Why it could happen? Imagine if you have very acidic formulation and if you are trying to inject, and blood pH is 7.4, the plasma pH, then very acidic or basic formulation can induce some pain at the site of action. You don't want to have any precipitation, and this is typically the problem for the poorly soluble drugs, because you try to use a co-solvent system. We'll see the details in the later part, but maybe the drug is very soluble into the solvent system, but the moment you're trying to inject what happens, the spore solvents, they get mixed with the plasma, and then drug will precipitate it out. If the drug is precipitating out, it will block the capillaries, and that will induce some side effects. We don't want to see that. Naturally, it has to be sterile. Tonicity I already discussed. You don't want to have any, pyrogenicity issue. The API or the excipients you're using, they must be pyrogen free. And we don't want to have any particulate matter there, which could block the capillaries or which could block the needle or syringes, for example. And then other factors we need to consider, they're like biopharmaceutical consideration, then solubility and stability. These are more CMC issues. So in our language, when I say CMC, it's a chemistry manufacturing control. It's one of the most important part of your dossier you will submit to FDA, US FDA or EMA. And then from the CMC point of view, for me, solubility and stability is very important. So from the concentration, you see that pharmacological response. Higher the concentration may lead to some toxicity. If it is lower, naturally you will not have any pharmacological impact. Then the desired onset of action and dose. So why this is important? Because anyway you are dealing with the parenteral formulation, so bioavailability is not an issue there because drug is directly in the systemic circulation. But what could happen, imagine you are developing a nanosuspension. So in that case, the first thing what drug will do, the drug will undergo into the solution because it will be insoluble state, and then that there is some lag, and that's the reason. Then desired onset of action, how fast you want, or whether you want some delayed effect. You will see some formulation to the market given by intramuscular route. They are nanosuspension based formulation, so the slow release of the drug takes place from the muscular depot, and that also gives the depot effect. You'll see a lot of anti-HIV formulations onto the market, especially in European market. They are developed like a long-acting parenterals, and that's how they try to give the chronic effect. Because for the HIV treatment, the patient adherence is very, very important, like you will see in case of TB. And that's how they tend to come up with this nanosuspension type of formulation. So once injected for 3 months, patient does not need to take any other medication. Dose is always an issue because for oral route, what happens, you are going to compress tablets or capsule. In that case, you can take as much as possible. That's not a problem. Of course, there are some limitations. You can't take in whatever quantities you want. But in case of parenteral, of course, you need to put that drug into the solution. And if the solubility is an issue, then high dose could be an issue. This is typically a problem with a lot of antibiotics. You'll see the antibiotics are always given in the gram quantities, 1 to 2 gram, maybe higher. And to dissolve that much amount of drug in a given formulation is always a challenge, because you don't want to also increase the concentration of your excipients. This is to be considered. Let's move to the solubility. Can you see my pointer here? Okay. In terms of solubility, especially when we are dealing with the poorly soluble drugs, so there are some approaches which we can consider, like use of buffers, salt formation, co-solvents, surfactants, other details also you see later. Then you also need to have the typical pH range. As I said before, extremely acidic or extremely basic formation may lead to some pain at the site of action. You may have some intolerance. We should also think of using the enabled formulations like nanosuspension, liposome. Stability, I said before that naturally you would like to have the shelf life of 2 to 3 years when stored at 2 to 8 or 2 to 8 degrees Celsius or room temperature. You don't want to have any degradation of your drug due to the oxidation or hydrolysis. This is to be taken care of, and to achieve that stability, and when you are designing formulation, you need to do some supporting studies in that case. So basically, what physiological consideration you can take once the formulation is developed? Before going into the clinic, you can always check if the formulation is inducing hemolysis. This can be done on the, let's say, the in vitro test where you can use RBCs. The tonicity can be determined. The precipitation issue can be determined when you are mixing with the stock or the organic part of your formulation with the aqueous part, the currently available injectable dilutes. That will already tell you if the drug is precipitating out or not. And if it is the case, naturally you have a chance to further optimize that formulation to rule out any hemolysis or precipitation issue. Other important aspect then from the manufacturing on handling support, FDA will certainly ask whatever formulation you have developed, because imagine any formulation to the market will come into the vial. What the nursing staff is doing, they will take some formulation out, mixing with the injectable dilutes, maybe saline solution, dextrose solution, or ringers lactate solution. but when they're mixing there, so naturally they should be compatible. You don't want to see any API precipitation there. You don't want your drug getting absorbed onto the infusion bag, infusion line. And when the formulation is being manufactured into the plant, you don't want to have any adsorption of the formulation onto the manufacturing equipment. You don't want to see that your formulation is extracting some part of the equipments or membrane filters that will lead some extractable leachable issues. So this has to be addressed. Then feasibility of terminal sterilization. This is more important for the drugs or formulations which you cannot really handle in the aseptic area. So in that case, you may have a chance of using terminal sterilization. Hemolysis, I think this is something we know, but I'll just quickly explain that when your formulation is not isoosmolar, I said before, the hypotonicity of formulation may lead to the hemolysis, and this is due to the disruption of RBCs. Is relieved, lead to renal failure. So this is a very important aspect which the precipitation I briefly discussed before. It might happen at the site of administration due to the rapid pH shift, due to the mixing of your let's say your formulation with the plasma, and this is to be avoided. How you can do that? From the formulation point of view, you may think of adding the polymeric precipitation inhibitors, which may take care of inhibition of precipitation. There are some other methods. Of course, you can always go for higher solubilization concentration, or you may think of some complex formulation, but this must be addressed. Phlebitis is something, again, the undesired adverse event when you are injecting the formulation. It may happen due to the presence of particulate matter, precipitation drug, or local pH effect. So particulate matter you can certainly take care of using the filters and aseptic processing. We have already discussed the precipitation of drug. Just as I said before, you can use polymeric precipitation inhibitors. Local pH effects, for that reason, make sure that your formulation is with the desired pH range between, let's say, 6 to 8.5, not very low acidic pH, not very basic pH. If you are trying to be with that pH range, maybe that is going to be very helpful. Pain at the site of action. So not only formulation, you may have some drugs like macrolide antibiotics, some excipients, which may lead to pain at the site of injection, and that needs to be avoided. In that case, then you can think of ruling out those typical drugs or excipients which may lead to pain at the site of injection. Typical example I can give you, I was using one weak base into the formulation, and at some concentration, that weak base was known to induce the pain at the site of injection, and this we only learned during the clinical trials. And naturally, when this adverse event were shared with us, and formulation scientists have the choice either to replace that weak base or just reduce the concentration of that weak base, so that you don't want to see that pain at the site of injection. Compatibility is very, very important. I briefly said to you before, but in that case, I would like to repeat, and I will go in more details. As said, you do have developed a parenteral formulation, maybe co-solvent based formulation, maybe by changing the pH, maybe nanosuspension formulation, or maybe liposomal formulation, but it's never injected as it is unless it is a bolus, because when you are going to inject the parenteral formulation, and it is given like a rapid bolus, like 3 to 5 ml, maybe up to 10 ml, or maybe there is an infusion which goes for, let's say, 90 to 120 minutes. But when it is infusion, you have to dilute that formulation with the diluent, such as saline, ringus lactate, or dextrose. So in that case, it's very important that your formulation, when mixed with that diluent, there is no precipitation of the API. The drug is not getting absorbed into the infusion bag. It is not extracting something from the infusion bag. When it is passing through the infusion line, again, there should not be any extraction from the infusion line. Same is true for catheter as well as needle. And that's what you would like to address into this compatibility studies. And FDA would really like to see that part. You have to do it, you have to mimic that clinical trial administration protocol into this compatibility to show that your drug is not doing, your formulation is not doing something to your infusion lines or infusion bags, and neither infusion bag or infusion line is doing something to your formulation. This is something we have to prove, especially for the clinical trials, for the when you are going to market the drug. Is very important to see. Imagine a drug is developed in US, and the diluent manufacturers could be different. Whatever saline solution you get in India could be different from US or Europe, or maybe in Asia Pacific. So we have to really make sure that all the sort of diluents are being studied into these studies to make sure that the drug is really compatible with the different diluents. And then same sort of studies you also want to do with the IV admission sets, inline filters and syringes to check that there is no drug adsorption or the formulation is not extracting something from these devices or the materials. Some more details regarding this, that when you are going to use the membrane filters, because for the aseptic filtration naturally you have to use the membrane filters, so then you have to make sure that the drug is not the formulation is not extracting something a filter, neither filter is going to block your drug, it's not absorbing any drug from the formulation. The same sort of studies you have to do with the packaging. Okay, with the glass vials you do not have any issues, glass is fairly neutral, but what if you are using plastic materials for storage, and also the syringes and of course you do have rubber stoppers there, so in that case you have to show that there is no absorption of APs onto these materials. And typically what you can do in that case, you have to store the formulation into this packaging material, store it for some time, and you have to determine the assay of that material by using HPLC. Appearance and pH will also give you some idea how stable is your formulation in contact with those packaging materials. Is it clear so far, or I'm running very fast? In case if I'm very fast, just tell me, I'll slow down just to make sure that you're not missing something. Can you just give me a brief feedback if it is clear so far?
It's okay, Yogesh.
Okay, thank you.
No
Thank you
issue.
for confirmation. So regarding the compatibility with the membrane filters, what you have to do normally, the solution you have prepared, either it's aqueous solution or organic solution, it is passed through the membrane filters. There you are going to 0.22 micron filters. It's a typo, it should be micrometer, not the millimeter. And you have to show the compatibility of the formulation with the membrane filters. And at the same time, you will also check that membrane filter is not absorbing any drug from your formulations. And there are recommended tests here, like microbial membrane detection testing. In that case, then product has to be passed through the filtration, and then you have to really check whether the filter is able to take out all the bacteria it's supposed to remove. In that case, filtrate has to be then incubated into the media to check that there is no microbial growth. In other case, the membrane compatibility, then in that case what you try to do, you try to soak the membrane in contact with your formulation and just verify then whether the membrane properties are damaged, whether your formulation contains any extractables coming from the filter. This has to be verified, but this data will be certainly needed. This guides you what type of filter you are going to use for your studies, and this is also part of your filter validation studies. The selection of container and closure, because the primary packaging is very important in case of the parenteral formulation, because it has several functions that it tries to protect your drug, because a final formulation must be sterile, so you don't want to have any free passes of the air or gas in and out of the formulation. Then you don't want to have any pyrogenic growth there, as it is a parenteral formulation, so it must allow the nursing staff to inspect the contents. Manufacturing feasibility is very important, that whatever unit we are going to use to manufacture, the primary packaging selection should allow to manufacture. Imagine I'm using an XYZ CMO in Austria to develop my product. They must have some setup to, let's say, manufacture the lyophilized vials. But in case if I'm coming with the liquid formulation to be stored at 2 to 8 with the infusion bags, then that will not fit together. So this prior consideration is very important in that aspect, that the primary packaging must be robust enough that it allows you to have transatlantic, trans-Pacific shipping wherever you are going to work, wherever you are going to use this medication, and that it should be convenient for clinical use. And when we are going to select this primary packaging components, then we have to consider the product stability, potency, toxicity, and safety. Now, for that purpose, you have to ask for all the possible documentation from your suppliers, because this will be needed during the submission. And here I tried to summarize, let's say, some important aspects when you are trying to develop the parenteral formulation. So depending on the drug's properties, like physiological properties, mode and speed of action desired, dose requirements, there are some considerations. If your drug is water soluble, If answer is yes, then naturally you just have to dissolve your drug in water and you just go ahead. But imagine if it is not soluble, then you are into this issue of poor solution. You need to consider some alternative options like pH adjustment, salt formation, because salts are more soluble than the free base or free acid. Use of surfactants co-solvent allows you to put drug into the solution. Complexing agents like cyclohexanes can be used to form a complex and to get into the solution. Having emulsion and suspension could be an option. When I say suspension, up to 5 micron is allowed for parenteral medication. That is one possibility. Lipid-based formations like emulsion, microemulsions, or liposomes can be considered. The non-aqueous oily vehicle, you will see some drugs like intralipid emulsions, the dosage form you can consider there. So all these options allow you to formulate the poorly soluble drugs for parenteral administration. Then once the formulation is ready, there are other aspects and stability consideration. If your drug is very robust, not undergoing any stability, naturally you are not worried. But if you have some stability issues, then you may think of some stabilizers like buffers, antioxidants, chelating agents, preservatives. But for that, you must know what is the mode of degradation of drugs. Once that chemistry is known, then it allows you to selection of particular excipients and you can stabilize the drug. But nothing is working in a liquid state to stabilize that drug, then you may have a chance of making a dry product. So in that case, you may think of just putting drug alone into the vial so that it can be stored for 24 to 36 months. So basically, it's a dry powder for a reconstitution. So what happens in that case, you come with dry powder. In another vial, you will have the liquid for reconstitution. It has to be just mixed prior to the injection. It has to be delivered. So you can rule out that. Lyophilization is another issue, but again, quite complex process, expensive, but depending on the stage of the production strategy that can be decided. Then having suspension, another issue, because the degradation rate in the solubilized state of drug is always higher than the, let's say, insoluble state. Then suspension formation could be another issue. Then biological consideration, we briefly touched this topic before, but again we can at least revisit that. Then the tonicity, hemolysis, precipitation upon injection, phlebitis, pain on injection, this has to be ruled out. And once this is addressed, then you go on addressing other issues we just discussed before. Then you need to see the compatibilities with the diluents, container closures, IV sets, installation filters. Immune stability also you need to determine for the microbiological stability. Feasibility of installation to be determined whether you want to go for aseptic processing and filtration or you want to go for terminal installation. Container closure integrity is very important to see, because at the end of the day you want your primary packaging to protect your drug for 24 to 36 months. So this was more or less the basic introduction what we can consider when we are trying to develop parenteral product. And once we know, then the options we have to develop the poorly soluble drug for parenteral medications are listed on this slide. One thing I can tell you, if nothing really works from this slide, it's better to tell your project manager or management that we should skip that drug, better not to develop, because as a formulation scientist, that this is maximum you can do to develop the parenteral formulation of poorly soluble drug. For today's talk, I'll mainly concentrate for the nanomedicine. So basically we'll be discussing the lipid-based systems such as liposomes, microemulsion, mixed micelles, and then the nanosuspension. So I think it's very interesting and promising strategy for the poorly soluble drugs, because it can really help to address the solubility issues where we are aiming to increase the bioavailability of the drugs. There is also possibility to reduce the side effects, because just to give an example, you do have the Taxol medication onto the market, which was always basically it's paclitaxel. Taxol was the brand, but it was always formulated using the antihistaminic drug was very important and necessary. So nowadays you will see Abraxane, which is a nanosuspension based formulation of paclitaxel, and there's a great reduction of the side effects. Just to give an example. Then it's also possible to target the drug as specific part of the body, because if you are able to let's say label your nanoparticles by specific ligands, so especially for oncology indication this is very important, then it's possible to target the drug to specific part. Depot effect. I think in the beginning I explained you that due to the low solubility of the drug, there's a delayed release happening from the muscular depot, and you may expect the depot effect from this nanosuspension based formulation. There are some advantages listed that you can go for higher drug loading, because typically you can even go for 5 to 10% drug loading. And said that 50 to 100 milligram per ml, which is quite high drug load for a parenteral medication. When you think of a similar formulation by using solution-based approach, it's very difficult. Low surfactant load, because for nanosuspension you hardly need 1 to 2% of surfactant. Imagine you are developing a microemulsion formulation, the amount of surfactant needed may be in the range of 15 to 20%. Chemical degradation is less because drug remains in the insoluble state. You are not using any co-solvents. The pH is not very extreme, and PK profile can be adjusted. So in that case, this is a typical definition of nano product. So in that case, you are expecting the particle size of the API less than 1 micron, and it is stabilized by surfactants, polymers, and mixtures of both. So what are the factors that influence the nanosuspension efficacy? So if the particle size is less than 200 nanometer, the solubility is quite high and it will produce immediate dissolution. But if the particle size is high, then dissolution of that drug becomes a rate limiting step, and then the PK profile will not be same. You need to adjust the zeta potential to achieve the better stability of that nanosuspensions. From the parenteral point of view, you need to make sure that the formulation should not be irritant, and whatever particles of the API you are going to achieve should not block the capillaries. This should be considered. And as it is a suspension formulation, the viscosity is always going to be higher than the solution-based formulation. So you need to consider the syringeability aspect, that the formulation must be syringeable. You can easily remove that formulation using the syringe. And some stability issues very important to consider. So I was too fast. The aggregation, also ripening, sedimentation, crystalline transformation, plasma stability. So aggregation and Oswald ripening we'll discuss shortly. Crystalline transformation, because the polymorphic form of the API is very important. You don't want to change the polymorphism of API changing the over a period of time. So this has to be addressed. And regarding aggregation Oswald ripening, we can just briefly follow this figure. So if your stabilizer added to the formulation is not enough, naturally due to the very high surface area of this nano sized APIs, they tend to aggregate, and once the formulation have aggregates, of course you do not have the stable formulation. If the concentration of stabilizer added is very high, what it will do, due to the high surfactant activity of that particular formulation, the drug will start getting into the solution. So you will have a situation where part of drug is into the solution, part of the drug is in the insoluble state. Then what happens, the drug will start precipitating out, crystallizing out, there will be crystal growth, and that's called Oswald ripening. You don't want to see that effect. But if you have the optimum stabilizer concentration, then there's no aggregation, there's no Oswald ripening. So I think it's very important. That's how you're going to optimize your nanosuspension. So you need to take the relevant formulation trials to make sure that the optimum concentration of stabilizer is used, and there's no aggregation as well as there's no Oswald ripening. There are some, let's say, not official guidelines, but from the formulation point of view, there are some considerations you should take into account for the selection of stabilizers. So what sort of stabilizers? If you're using ionic stabilizers, naturally it will try to inhibit the aggregation by electrolysis repulsion. In case of non-ionic surfactants, they try to stabilize the formulations by steric repulsion. Surface energy consideration should be taken into account, because some polymers like cellulose or PEPs, they try to stabilize the formulation by the virtue of hydrogen bonding, because they do have hydroxyl groups present onto the let's say form this non-covalent hydrogen bonds that will certainly help to stabilize the formulations. The hydrophobic moieties of stabilizers are very important, because it tend to get absorbed onto the surface of these hydrophobic drugs, and once they are getting absorbed, like layer of onto the That's how it is trying to prevent the aggregation. It's trying to between the two like molecules of the drug. If you are using surfactant like polymer, poloxamers, 280, basically these are the surfactants. So, and you are using this surfactants as stabilizers. So you don't want to use those surfactants above the critical micelle concentration, because if there's a high concentration of surfactant, it will lead to solubilization of drug. You don't want to see that. As we discussed before, it might lead to the post-wall ripening issues. If you're using food grade proteins as the isolation must be considered there. And whatever stabilizer you are using, that your drugs should not undergo solubilization into that stabilizers. So what are things you need for to develop nanosuspension formulation? So you need like a tonicity modifier, because at the end of the day you need iso osmolar formation. Cryoprotectant might be needed because you have to. I mean, it's only needed if the end product is lyophilized. If you are using nanosuspension as it is for long-term storage, you don't need a cryoprotectant. Buffer in case a lot. Co-surfactant is really not important, but it really depends. Maybe 1 to 2% of co. So there are different suspensions. You can think of using bottom up, top. The HPS technology is there. So the bottom up basically is the precipitation technology. So what happens in that case, you're trying to dissolve drug in a solvent, and then another anti-solvent is added, so the drug is precipitating out. But this is a controlled precipitation, and during this controlled precipitation, and the nano product. So it's simple and low cost. You low particle size, but the octane system, you may not be able to get rid of all the organic concentration. So you might have some organic solvent. It's not universally acceptable. That in the top-down approach, basically you're going to use the mills. So typical nano mills, you may think of like Elan's nanomill is there. You have some niche mill. The zirconium beads are used there, and basically the drug, the aqueous dispersion of the drug is subjected to the milling beads. You will see a lot of commercial. So it's a low energy technique, batch to batch. But there's a risk you may have some residues from the milling media, which is that's the reason I mentioned the zirconium medium stainless steel. The process is slow because you typically need 3 to 6 hours of milling time. Sometimes it's not quite stable. The high pressure homogenization is another aspect you can consider. This technique is universally applicable, easy downstream process. So in that case, what you do normally, the aqueous dispersion of drug is subjected to the high. You have this formulation of high pressure and high energy system, and that's how the nanosizing takes place. You have this typical technology, different processes to prepare nanosuspension. These are all commercial technologies, so commercial success is there. So you can consider one day if you need to develop the nanosuspension of poorly soluble drugs for parenteral medication. These are the options you have from the nanosuspension side. For this, you may think suspension. So APIs with low lock P, the partition coefficient to have stable nanosuspension region is that in that case chances are quite high. The API may not characteristics, or it may form some lumps or gel. So this has to be avoided. The APIs with low enthalpy values could be considered poor candidates, and also there are some consideration for the approach you are using. So for the bottom up, we discussed before, it uses solvent to solubilize the drug. But imagine if your drug, which is not soluble in any of the class 3 solvents, because you can't use at the end you want to remove all the solvent, but it's always not possible. So there you have to consider which solvent you're going to use, and if your drug is not soluble in that solvent, it will be difficult. In case of top-down approach, like a nanomilling, then you need to consider that the API must be dispersible into the vehicle you are going to use. Some drugs, they just tend to sit onto the surface. Some drugs, they are just forming a gel. Then you can't use top-down approach. And this typically mechanism you can see here, what happens when you go for the nanomilling option when you want to stabilize your drug. So basically, drug is subjected to the grinding media. That's how the nanoparticles are generated. If there is a good presence of stabilizer and stabilizer working is very well, you have very good control of the suspension stability, but the stabilizer is failing, then the particle aggregation will take place, and that leads to the unstable nano suspension formulation. There are some case studies I tried to take from the market using this I'm sorry, approach. So you have the formulation Aristida initio, and this formulation basically extended release formulation for the aripiprazole loroxil. It is given by intramuscular route, and as it is a nanosuspension formulation, the composition is listed here. So basically, it's given by intramuscular route. It forms a depot there, so the particles will be resting into the muscle, and due to the slow dissolution of the drug, you can expect the depot effect for 60 to 90 days. Another example for this parenteral medication is Invega Stena from J&J. The composition listed here, these are typical like polysorbate 20, PEC 4000 type of stabilizers they are using. We do have some blockers to adjust the pH. Again, it's the same, it's produced by nanocrystal technology from Elan. The particle size is less than 400 nanometer. And then, as said before, due to the low solubility of the drug, it forms a depot at the site of injection, and the delayed release is possible. So basically, the nanosuspension, they are thermodynamically unstable. So then that's the reason we are using stabilizers there. So you need to focus on typical aspects in order to have the stable nanosuspension. So we need to take care of aggregation, crystal growth. Polymorphic form must be monitored and kept constant. You don't want to have any sedimentation of population. Chemical instability point of view, there is no greater risk. Maybe for degradation, one aspect you can look, if it is photosensitive, maybe the umber color type of content should be considered. Oxidation hydrolysis risk is low, but nevertheless should be considered there. How you want to address the stability issues? So that really depends what kind of degradation you have. I mean, there are two issues: physical stability, chemical stability. So if there is physical stability issue, aggregation, then you need to find out whether it's a problem of osmolar tightening or it's a problem of aggregation, and depending on that, you can really decide then which stabilizer has to be used at what concentration. That will really help you to optimize the formulation. And from the chemical point of view, once you know whether it's oxidation, whether it's hydrolysis, depending on that, you can consider selection of the stabilizers. You can stabilize it. Briefly, I discussed before, often the formulations are very unstable in the liquid state, but if you try to make them dry by using solidification or drying technique, the risk or the chances of stabilizing that formations are very, very high. Chemical stability already I discussed before, so I will not go into the details, but in a nutshell, you can just think of the risk of the mode of degradation of the drug, and then what typical excipients you need to use stabilizer. As example, I can give a lot of drugs that undergo oxidation or hydrolysis. Maybe you can think of using the complex agent like cyclodextrins, because in that case, what happens, the labile part of drug might go into the cyclodextrin cavity, and that's how you can impart the stability for the desired formation. Of course, the solid or concentrated product is more stable than the liquid formulation. Packaging consideration is very important for the photosensitive drugs. And what happens when the nanocrystals are administered by IV route? So typically when you have the particle size less than 200 nanometer, I said before briefly, it undergoes quick dissolution and then it is available for the system circulation. But if they are higher, let's say more than 400 nanometers, then they're not undergoing dissolution, they will be taken by macrophages, and then that's how we also call RES uptake, reticular endothelial system uptake. Then you will have high part of your drug getting deposited into the liver, and then you are landing into the safety issues discussion with the FDA. So this is very important that you need to carefully consider the particles of the drug into the final formulation. So this was more or less for nanosuspensions. Let's discuss briefly. So In that range, you can think of developing liposomes, microemulsions, mixed micelles, intralipid emulsions. And this is very interesting option for the IV administration of poorly soluble drugs. And typically, if your drug have very high LOCP, like cyclosporine, high LOCP indicates the lipid affinity of the drugs. And if your drug is getting solubilized into the lipids, you may think of developing lipoemulsions or microemulsion. But it's very important that API must be stable in contact with lipids. Often these lipids, they contain the residual oxidizing impurities, and that may lead to the degradation of the API. Important aspect that concentration of surfactant should be monitored quite closely. Typically, if you see any microemulsion composition, you will always have 15 to 20% of surfactant there, or maybe more than 10%, and sometimes it's not very tolerated very well. So this is to be avoided. Normally, the liquid formulations do not induce any osmolality, so this is to be carefully considered because you need then suitable dilution, diluting or suitable diluent to dilute. So let's start with microemulsion. Basically, these are very thermodynamically stable system once it is mixed, and these are like mixtures of oil, water, and surfactant and co-surfactants. Very easy to formulate. You don't want to impart any energy. Basically, processes that you must be knowing. You dissolve drug in an oily vehicle mixed with the surfactant, co-surfactant, or like typically SMETS part, as we say. And once this SMETS part is mixed with the water, you get a microemulsion system. So it's very popular, at least for oral route. There's a lot of research going on in India and abroad. There are a lot of formulation to the market. Typical example is cyclosporine. But for IV, for parenteral purpose also, you can consider this formulation. And this gives one possibility to deliver the poorly soluble drugs. The typically microemulsion formulations for the parenteral would consider lipid droplets. It contains the lipid droplets in the range of 10 to 20%. It has osmotic agent and emulsifier. If it is a multi-dose containers, you need to also consider the addition of antimicrobial agent. From the market, the typical example for microemulsions is propofol injections. So earlier formulation was based on crimoporial and ethanol. But okay, we know that due to the crimoporial, there are some histamine-induced reaction. Later, they tried to replace crimoporial with ethanol. And finally, they came up with the microemulsion-based formulation for formulation is very well tolerated. The safety. And on this slide, you see the different microemulsion-based commercial products for parenteral administration. So you get type of excipients they are using, what is typical drug loading, what kind of diluents they are using. So this will guide you, and if you want to develop the microemulsion-based products for the parenteral medication. Mixed micelle is also a very important aspect. So in that case, the typical process you see the drug and the surfactants as well as the stabilizers are dissolving organic solvents. Organic solvent is evaporated. You have like thin film, which is then hydrated with the buffer, and you get this mixed micelle type of delivery system. This is very important case study I tried to use during my Swiss time. So there was a company called Forest. They had this ready-to-use kit to prepare the parenteral formulations for early trials. Where you don't want to spend a lot of time to develop a formulation because these are investigational compounds. You never know if it is going to work or not. So it's better to have something ready. So basically, they provide this kit, which is basically the lipidic dispersion in the buffer. It contains 100 milligram per ml. Like ethanol, depending to 20 milligram per ml, and then this ethanolic solution is added to the this phosphorus. Initially, as you will see, the drug might precipitate out, but upon further mixing, it's possible to encapsulate the drug within the phospholipid, and then you do have the formulation ready for the IV injection or intramuscular injection. So I think it was very, very important to have this medication ready or to have this formulation ready for the preclinical dosing where you don't want to put a lot of API, the time and money. And it was quite useful. Unfortunately, this is no more available into the market. Some case studies from the literature where they have used the mixed micelles for curcumin. As you can see from the graph, the free curcumin undergoes quite significant degradation upon the parenteral administration. But when they had mixed micelles prepared using two different types of stabilizers, the stability was quite good into the plasma, and then this is one liposomes, very important dosage forms. So these are basically the spherical structures. You do by the lipidic bilayer. So what happens, the water soluble drug will go into this hydrophilic core. If your drug is lipid soluble, it will reside into this hydrophobic layer. Depending on the API properties, you may think where you want to really put your drug into these bilayers. And for the encapsulation, there are other aspects like what is locking up your drug. There are different methods available to load the drug, and depending on that, it impacts the size of liposomes you are going to have. There are also labeling aspects. You'll see a lot of pegylated liposomes onto the market, and pegylation is mainly done to improve the circulation time of liposomes. If without pegylation, you may have some RES effect, as we discussed before, that the liposomes will be taken out by the RES systems, and then the overall circulation time is low. You'll see a lot of liposome-based IV formulations into the clinical trial, and typically for macromolecules like peptides, proteins, antibodies, this approach is very useful because it's very complex and expensive technology. So if you want to use for small molecules, of course you can use, but then you need to justify the overall cost of the operation. From the market, I think the doxorubicin is a very important example. Doxorubicin anthracycline anti-cancer drug. If you see the mode of action, it inhibits the DNA and RNA synthesis, and it acts through the inhibition of enzyme topoisomerase, basically. So what happens in this case, this positively charged doxorubicin has very high affinity for the protein called cardiolipin. This cardiolipin is present in the heart tissues, and the cardiolipin is negatively charged. So this positively charged doxorubicin binding to this negatively charged lipid, and due to this interaction, there is a irreversible congestive failure occurring, so leading to even death. So the cardiotoxicity is very high for this doxorubicin formulation. And that's where they tried to come up with the liposomal formulation to overcome these toxicities. Into the initial trials, they tried to use the negative charge liposomes, where drug was, let's say, they tried to achieve the drug loading by using lipid hydration method, but the clinical trials were failing because the drug release was quite fast. The liposomes were cleared by reticuloendothelial system. Then they tried to use the remote loading because it's a different loading method where they tried to improve the drug loading. Even the circulation time was improved by, let's say, the pegylation, and that's where they came up with the market product like myosin and doxil, and the loading method was different, like pH or ammonium gradient. And on this slide, you see the difference between the free drug and the marketed liposomal formulation. So as you see here, myosin is coming with the pegylation. And if you see the PK profile, the AUC for the myosin is significantly higher compared to free drug and doxil. Even the half-life is improved for myosin due to this pegylation. The incidence of side effects is also very low for the liposomal products compared to the free drug. And same is the case for the doxil. So in a nutshell, the circulation time is high. The incidence of side effects is very low for the liposomal products. On this slide, you see some marketed liposomal products. As said before, these are mostly into the oncology systems. They are also for the anti-infective sections, but the drugs they are using are mostly the complex molecules which are quite difficult to be formulated by conventional approaches. So the due consideration has to be given then whether you do have other alternative options, because the technology is quite complex, quite expensive. So if needed, of course you can go for that, but then there should be a strong rationale. With that, then I'd like to conclude. So formulation development of the parenteral nanomedicines is not very simple. Is often not limited to simple choice of excipients process, but there are some typical aspects we need to carefully consider so that we don't want to see any complex into the future. We need to carefully consider what are the typical API stability issues, what type of excipients we're going to consider, what is the compatibility of API and excipients or the infusion line and the infusion bags, etc. In terms of process, what are the complications? If there is any primary packaging compatibility issues, what are the end user requirements? This has to be carefully considered and addressed. For the poorly soluble drugs, it's always not easy to formulate, so we need to carefully consider what are the options we do have to select the enabled formulations to come up with the successful parenteral delivery options. So with this, I would like to conclude my today's talk, and if you have any questions, I will be happy to answer. Hello. Hello. Can you hear me? I can't hear. Dada, you're on mute, I think.
Hello. Yes,
now
yeah. Thank you, Dr. Yogesh. It is always my pleasure to hear you. Every time I hear, I get some new information and new dimension of research. Thank you so much for your informative and insightful presentation. I have thoroughly enjoyed your talk, and same must be true for the participant for sure, I think. You have very lucidly elaborated all the technical aspects, starting from pre-formulation and other consideration for developing parenteral nano formulation, and nicely described the packaging requirement and its importance and other details, which we very often skips. And to be very frank, nowadays we all focus on novel drug delivery route, like transdermal route, mucosal route, nasal route, etc. But we neglect very basic and useful parenteral route, which is still the first choice for emergency and also for some critical treatment like cancer therapy. I'm sure your speech will generate new interest to our participants to do some basic research on this aspect. So, Dr. Yogesh, with your permission, I would like to raise some questions put by the participant in the chat box. But let me announce, if any participant is interested to ask question directly to Dr. Yogesh, we will unmute him or her. So first we will take the consideration of any participant if he is interested. Okay, meanwhile, let me ask one question from Dr. Archana Kalekar. Yes, who is this?
Yeah, good afternoon. This is Bappaditya Chatterjee from Mumbai. Yeah, may I?
Yes, carry on.
Yes, thank you, Yogesh sir. It's good to hear you. I'm hearing you second time. It's very nice. Yeah. Just I want to ask your opinion, like what we do in academic research, mostly for parenteral preparations, we develop the formulation, and then we try to think about some packaging and we fill it and go for sterilizations. That the thing. So I just want your suggestion that at which step of development we need to take the packaging material into considerations, at the very beginning or after developing the formulation.
Thank you, Bappaditya, for being there, and it's equally pleasure for me to hear you. Yeah. So regarding your question, I think you are rightly focused. So as a first part, you will just try to see what is your prototype, what kind of excipients you are going to use, what is the aqueous part it is going to tolerate, what is the drug loading you are able to get, whether there is no precipitation of the drug. Once you have addressed all these aspects, so primary packaging you must consider before you are going for, let's say, the aseptic processing all. Because what happens, imagine if you have not selected your primary packaging, you finish all your formulation optimization, you even finish your aseptic processing, etc. And then you realize, okay, if your drug might getting adsorbed onto the rubber closures, maybe it's not compatible with the glass you're using, because nowadays you will even see you get siliconized glass, because often some proteins are getting adsorbed onto the type 1 glass. Same is the case for rubbers as well. And if it is a plastic packaging, in case you will see some plastic packaging for the liquid formulation. So, and if this is a plastic packaging, naturally you need to address the issue of adsorption. So all these aspects must be addressed before you are going for the very final formulation optimization or aseptic processing, because that will assure you, okay, you have made a right choice of primary packaging. Another aspect I addressed during my talk, water okay, it's more industrial aspect. Once you are going for manufacturing your formulation, what kind of line you are going to use? If it is a lyophilized product, naturally then whatever life setup you are going to use, it must be able to handle the glass vial you have selected, whether it's a 10 hour vial, 20 hour vial, what type of the rubber closures you are going to use. So, and once you have already selected that, it allows you to go for that particular manufacturing option. Otherwise, then it will be little difficult or late.
Okay. Yeah, I got your point. Yeah. Okay. Thank you, sir. Thank you.
Thank you, Dr. Yogesh. Now we have one participant named Dr. Archana Kalekar. So she is asking, I am a material scientist and have some expertise in synthesis various nanomaterials like gold, silver, titanium dioxide, copper oxide, etc. And even quantum dots as well. So I want to do collaborative research in drug delivery and drug targeting. Hence, I want to know what are actual expectation from pharma researchers will have from material scientists like her related to nanomaterial.
Right.
Like which are most useful material properties and nanomaterial.
I think it's very important question. So I think in terms of expectation, I mean, you need to fulfill the regulatory expectations. So if they would like to see whatever material you are going to use to make nanoparticles, you should be compatible with the, let's say, physiological system. It must be biodegradable, because see, if it is injected into the body, either it has to be eliminated from the body, or it has to be biodegraded. You don't want to have any accumulation of that material within the body. This has to be addressed from the material point of view. That material should not induce any toxicity, so it must be on the approved excipients list. If it is a new excipients, naturally you need to generate a lot of toxicity data to FDA. Okay, your material you're going to use to produce nanomaterials is not going to induce any toxicity. In terms of size, then as I said before, below 200 nanometer it goes quite immediate dissolution. Then, but if it is higher than 400 nanometer, then there's some encapsulation by reticuloendothelial system. There's a liver deposition. It may induce some toxicity issues, and only by generating the data you can prove to the authorities that material or the formulation you have developed is acceptable. I mean, whatever verbal argumentation will do, there are no standard set of guidelines for nanomedicine, at least, that I can really tell you, okay, you have to do this and don't do that. More you study, more you will generate the data, and that's how you can argue with the authorities and you can justify the potential product.
Thank you so much, Dr. Yogesh. And I think that addressed the question raised by Dr. Archana. Now we have one research scholar from our department, Shivani Pannu. She is asking, what should be the ideal properties of a nano carrier for parenteral administration?
Yeah, I mean, in terms of properties we discussed in the beginning, the final pH should be acceptable for the IV or intramuscular administration. You don't want to have extremely acidic or extremely basic pH. Osmolality should be acceptable. You don't want to have any hypoosmolar or hyperosmolar product. In terms of particle size, as I tried to answer the earlier question, if it is below 200 nanometer, it will undergo immediate dissolution, and that will give you, let's say, immediate dissolution. But if you want to have the depot effect, I tried to provide some case studies in my talk. That there are some long-acting parenteral products which are giving the effect for 3 to 6 months even. So in that case, you can even think of increasing the particles to 400 nanometer. Maybe you can even go up to micron. You do have PLJ type of microparticles into the market, and they are even working up to 6 months. So depending on the PK profile you want to have, you can really fine-tune the particles as if you're nanomedicine. At the end of the day, you don't want to use any excipients which may lead to any toxicity issues. The drug which might lead to oswald ripening, or there should not be any aggregation of the nano-size APIs which may also lead to the precipitation issue. So these are more or less the expectations from your nanomedicine for the parenteral administration.
So,
Hello.
Dr. Yogesh, yes, carry on. Yes.
Hello. Dr. Yogeshwar?
Yeah.
Am I audible? Yeah, I am Dr. Sushma from Dipsiru. It was a very wonderful talk. There are the certain.
Madam, you're on mute, I guess.
Sorry. Yeah. Am I audible?
Yeah, now I can hear
Yeah.
you.
I am Dr. Sushma and I am from Dipsiru. It was a very wonderful talk. But there are the few questions regarding this parenteral administration of nanomedicine. The first thing is, as you mentioned, that maximum nanoformulations, they are the pegylated or the normal, isn't it? Because lipidic formulations, they are generally taken up by the macrophage or RES uptake. So basically, they are the pegylated. But since we are in the actually in academics, we generally do the basic research, and I very well accept the fact we are very far from the industry's expectation. But in the research, so many I have developed the formulation for the targeted based delivery system, where we found there are the certain receptors which are overexpressed, especially in the cancer and all. But still in the market, there is, except the antibody based targeting, there are no other ligand based targeted products are available. What is the reason? Are they not stable in the inside the human body? For example, now in the case of breast cancer or in the colon cancer, for example, CD44 receptors are overexpressed. Everybody knows. Now in the case of breast cancer, there are so many receptors are overexpressed. So except the antibody, there are other preparations are not available. Even the ligand is very cheap and it can be affordable, but it is not acceptable by the industry people.
So thank you for the question first. So I'm not sure if I have totally understood. So I think we are trying to discuss 2 to 3 aspects here. So the pegylation is more important to improve the circulation of the liposomes.
Yes.
So maybe for nanosuspension, I don't need a pegylation because inherently it is an insoluble drug. The dissolution will really drive the bioavailability of that drug. But in case of liposomes, there's a tendency that this will be cleared by the RES system. And to avoid that RES something, you are going to pegylate. That's just first question. I mean that will help at least first part of your question.
Yes. But after pegylation, is there any chance that the drug will release through en route during its long circulation? Is it possible? Maybe there are some amount of the drug may be leached out or leakage. Yeah, or that may be assured before going for a clinical. That must be, I think, assured. During their long circulation, are they stable?
Yes, naturally. I mean, that is the reason, right? You're performing pegylation to make sure that there is no encapsulation. But when when you say stable, overall aim is that during that circulation, the liposomes also releasing the drug. You also need to have the release of the drug during that time frame, right? And depending on your drug loading and depending on the structure of your liposomes, what kind of lipids you have used, what kind of stabilizer you have used, that will really drive the release of the drug from the liposomes. And naturally, then when you do PK studies, you get that kind of information, whether it's a rapid release, whether it's a delayed release, whether it's an intermediate release, and based on that you will fine tune. But in that case, the driving mechanism is the release of the drug from the liposomes, not the RES uptake. Pegylation you are just using to block that RES uptake, then the varying factor is release of the drug from the liposomes.
My second question.
Yeah, and in terms of the targeting, I certainly agree with you that whatever research is happening is more or less is really in the basic research, that what kind of receptors they are trying to target, what kind of ligands they are going to use. But if you see like commercial products, there are not many onto the market which are really, used based on this targeting, because as you clearly said, the level of receptors being expressed on the cancerous cells is never consistent. It may vary from patient to patient, so it's too risky if you are trying to develop a product which is based on the targeting or ligands, and it may not
Okay.
be working on the different patient population. But nevertheless, if you see, a lot of research is going on onto the ADCs, the antibody drug conjugates, and
Yes.
that's mainly driven by the presence of the receptors effective, more let's say the working or yeah, functional compared to the typical ligands you are going to use. So I think ADCs are working quite well, and this is typically true for very potent drugs, because the in that case the dose of drug is so low, even in microgram. And
Yes.
that's the reason then they are going to use the ADCs, that you don't want to see any non-specific effect due to that drug. The drug is especially going to the receptor where it has to work, not into any non-specific receptor.
Okay. And last question. Uh, what are your comments? As in the academic institute, we generally use the rat or the mice model. So can we correlate the result what we are getting from the animal is so small, and then we are going how they reliable? So whatever reading or preclinical studies, in preclinical studies, whatever we are getting, what do you think? Is it a perfect model, or there is some big model we can use for a, if we want to go further for the clinical trial and all?
Yeah, I
So
mean,
are they reliable?
from the efficacy point of view, if you really
Yes.
want to see a proof of concept in terms of pharmacological efficacy, so you see a lot of even oncology POC studies, the preclinical studies are done on mice. So in the
Yeah.
mice data should work well to at least to confirm that whatever concept you are using to improve the efficacy is working well in the mice models, and you have at least good chances to build your theme that this might also work into the higher animals onto the humans. But when it comes to the PK profile, the pharmacokinetics, you can't really
Yes.
translate the 100% results from mice to rat, rat to dogs, or dogs to the humans. So
Yeah.
you get some basic idea, because you are always going to compare the free drug or the commercial, the conventional formulation against your delivery system. You get that kind of feeling, or you get that kind of information how your delivery system is going to work. You can always optimize, but once it is optimized, the aim is always to try into the highest species to make sure if the results are consistent or reproducible.
But they do in the large animal model in industry.
Hello,
They do they
can you still hear me?
perform the study in the large animals after performing in the rat or mice?
Sorry, I missed 2 to 3 lines. I don't know
No, no,
what.
no. I am just asking after performing the studies in the in the rats, do they go for larger animals?
Yeah. I mean, if I see the development from my point of view,
Yeah.
before you go into humans, the FDA would always like to see the data on the rodents and non-rodents. So typically,
Okay.
you start with the rats, and then you have to go to the non-rodents. So either it's a dog or monkey. It really depends on the your metabolic profile and the target human dose, which species you would like to see. But before going to the humans, you must generate the toxicity data into the higher animals.
Okay. Okay. Thank you so much. Thank you. Thank you so much.
Thank you, Dr. Yogesh. Any other participant? You could ask question. You can raise your hand. We'll unmute. So I think don't have. Dr. Yogesh, there's one basic question from Mr. Anis Pradhan. I think you have already discussed first, but in case you want to shed some more light on this, he's asking what are the different quality control parameter for nanosuspension?
Yeah, for a nano product, I think it's very important. The particle size is most important. That particle size must be reproducible, and that should be part of your specifications. So from the formulation point of view, this is most critical. Of course, there are some additional subsets to if you have to do for presence of visible, subvisible particles, and there are some specifications, and then you, every time you have to make sure that the typical specifications are passing. Imagine the D90 is 200 nanometer, but what is our D100? So if you see the pharmacopoeial limitation, you don't want to see any particles above than 5 micron. I mean, you can see, but there are some limits. As per the limits, then what are the particles in the range of 25 micron? So I don't have all those numbers by heart, but if you see the USP or European Pharmacopoeia guidances, you will see what kind of particles they would like to see into the final product. And then there are some traditional QC tools in the pH, osmolality, the appearance, etc.
Okay, okay.
So, can I ask the last question?
Yes, madam,
Hello?
please.
Hello.
Yes,
Yeah.
ma'am.
Just can you make us some light on the micelles, this mixed micellar system? Have you worked on that? How they are going to be formed, polymeric micelles or micellar?
Yeah, I can share the screen. Maybe I had few examples.
Actually, I missed that slide actually. How they are going to be formed below CMC?
Micelles are different compared to your standard micelles. So the question you asked above CMC, below CMC, that's typically applicable if you're using polysorbate ET alone or poloxamer alone. In that case, what you are going to do, you are trying to add the higher concentration surfactant to the water. And then if the surfactant concentration is so high, that drug will be encapsulated into the micelles and you get a micellar solution. In that case, what's happening, you're trying to use the stabilizers as well as lipids. You're going to dissolve in organic solvent like whichever, like class 3 solvent allowed, and dissolving the API. You're evaporating the solvent. You get a thin film there that is being hydrated with the buffers, and once it is hydrated, you do have the encapsulation of the drug prepared this using mixed micelles. The reason it's called mixed micelles because micelles formation is happening due to the combination of two types of stabilizers, surfactants, phospholipids you're using.
Okay, okay, okay. Okay, thank you so much.
Okay, thank you, Dr. Yogesh. And can I take one more question?
Yes, please.
One question from Mr. Dhanraj is asking, how can we improve the zeta potential for the development of solid lipid nanosuspension, since it is the major factor to consider?
I mean, it depends what you say improvement. Most important, first you should know at what zeta potential value your formulation is most stable. Of course, there are some recommendations. You need to have typical value of zeta potential to stabilize the system. But at the end of the day, you see where you don't have any aggregation, where the drug is not really coming out of the solid lipids, it is not remaining on the surface of solid lipids. So by changing the surfactant concentration, the surfactant will mainly drive the zeta potential of your system. In case of solid lipid, you are mainly using the solid lipid, which has to be melted. Then you're going to dissolve the drug into that molten lipid. And then when you are adding this molten lipid to the water, you do have the presence of surfactant, which will really control the nano sizing of solid lipid. So that particular concentration of surfactant, and of course the process of agitation you are using, it will also have some impact on the overall zeta potential. So by studying this process parameters, the surfactant concentration, type of surfactant you are using, it really help you to optimize the potential of your system.
Thank you so much, Dr. Yogesh. Now I have one question for you. Let's say we have developed one parenteral formulation with nano carrier. So is it mandatory to submit also the toxicity reports of that nano carrier to the regulatory authority?
Yeah, they will certainly ask for it. It's not for nanopore, for any formulation. If it is a new drug, imagine if it is a new drug, you will need a toxicity data of that drug. If it is a generic drug, you can always refer to the established toxicity data as long as you are not changing the drug and route of administration. But in that case, imagine you are developing a nano product of a approved drug, and if first time you are developing that nano product, then in that case authorities will ask the safety profile of that drug, because this nano product will change the PK profile of that drug. Either the absorption, or in that case if it is parenteral, the bioavailability might be higher, or maybe it's delayed, and what impact it has onto the efficacy of drug. And this is how you are going to prove into the animal species in the form of toxicity to prove, okay, up to that dose you did not see any adverse event, There's no toxicity, and based on that, you will project your human dose for the clinical trials. And that's the reason they will ask you to show the toxicity data.
Okay. Thank you, Yogesh. Thank you very much. It was such an interesting interactive session. And Dr. Yogesh, we have something for you. As a deep sense of gratitude and your appreciation, we are presenting you this certificate. This is for you. We'll post you to your address. So once again, thank you so much for sparing your time from your busy schedule.
No problem, Dada. For you anytime. Thank you. It was my pleasure.
Okay. Thanks a lot. Thanks a lot.
Thank you all. Thank you for the attention. I really enjoyed this session. Very interactive. Thank you.
Thank you. So dear participants, now we come to end to this session for